Okay.
Okay.
So today we're going to cover
Did we start?
meiosis. Yes. Okay, tell me what you know about meiosis.
Oh, me?
Both of you. Brainstorm.
Okay, so it starts with anaphase and then prophase.
No.
No, anaphase, metaphase,
No, no,
prophase.
no.
Oh, prophase,
Yes.
metaphase, anaphase, telophase.
Yes.
Yeah.
And what cells do meiosis? That's the key thing. We have two types of cells in our body, the somatic cells and the sex cells. Which ones do mitosis, which ones do meiosis?
Meiosis is sex cells.
Yes.
And then mitosis is somatic cells.
Yes. Okay, and then one thing that the textbooks don't really tell you, but that you have to know is germ cells are diploids. But the gametes that come from those germ cells are the haploids that do meiosis. So in an organism, the germ cells do mitosis and create lots of germ cells, and then those germ cells do meiosis to create the gametes, like sperm and eggs. You know how we learned about spermogenitus and oogenesis?
Oogenesis.
Yeah, it has like you have the gamete, I mean the germ cell, and then that germ cell does meiosis to create the egg, and you'll learn that next year. Okay, so now the best way to remember this, and Dr. Brooks taught me this, prophase, think about the word prepare. Metaphase, riddle. Anaphase, apart. Telophase, tidy up. And they all start with the same letter, so it's very easy to understand, okay? Now, since we're starting with a whole diploid cell, like 2N, each cell has a pair of chromosomes from each of the parents. 23 from the one parent, 23 from another parent. And if you can see here, I've shown you that you have one number 1 chromosome from one parent and one number 1 chromosome from another parent. The same with the 2 and the 3. So you have a total of 46 chromosomes, okay? Now what happens is, it's during meiosis 1 that the ploidy number goes from 2N to N, from diploid to haploid, in meiosis 1. So meiosis 1 is nothing like mitosis. Do you need something?
Yeah, I just want to ask you, how do you represent standard deviation?
I have no idea.
Oh.
Wait, let me see.
I'll spell, yeah.
You wanna? Where do you wanna represent it?
She told us to just represent it on the graph.
Then just like, can't you write it here? Like that's how we would write standard
No, I
deviation.
mean, is this right? Like,
No, this would be this. I think that these are error bars.
yeah, error bar.
Yeah.
So I'm not sure if these can represent standard, but like that would, because if this is the average, then that's how much it deviates from the average. So I
So
think
it has
that
to be
makes
the
sense.
same amount on.
It has to be like, it has to show where the average is and then how much it deviates from the average.
So isn't? Oh.
Oh, is this already averages?
Yeah, this is already averages.
Oh yeah, so then this would work because this shows that if this is your average, then it's deviated
It
that
deviates
much.
between them.
Oh,
But-
so this is right?
Yeah.
Okay. It doesn't have to be the same length from the mean, right? Not always.
Not always.
No, not always.
No.
Okay, so now during prophase, it's the same thing as mitosis in the sense that like the nuclear envelope starts to degrade, you know, the chromatin condenses, and you're left with
So with meiosis,
either way,
meiosis. There's meiosis 1, the meiosis 2,
Yeah.
and then meiosis 1. That's when it goes from diploid cell to haploid
Yes.
cell. Yeah.
And then in the second part, it's identical to mitosis. I normally
The
use
second part of meiosis is identical to mitosis.
Yeah, it looks a little different, but the processes is the same. And like a lot of people on the quiz, I mean, I've got this question wrong on the quiz when they ask you in which meiosis does the ploidy number drop, and everyone's like, oh, meiosis 1's like mitosis, so obviously it drops in meiosis 2, but that's not the case. And like I wrote here, meiosis 1, 2N to N, meiosis 2, same as mitosis. So
What is mitosis though? Like what happens?
Mitosis is the same thing, prophase, metaphase, anaphase, telophase, but you're left with 2 identical cells. In meiosis, you're creating 4 different cells.
And then in meiosis 2, you're left with 2 identical cells.
And 2 identical, 2 identical. Or unless crossing over has occurred.
Which happens
In prophase 1.
1.
Yes.
Ah.
Oh, yeah. Okay, so let's do prophase 1 then. Okay. So in prophase 1, the first step is chromatin condenses, the nuclear envelope degrades, like in every single prophase, okay? Homologous chromosomes stick together. A homologous pair is when you have like chromosome 1 from the parent, A, and chromosome 1 from parent B.
And they connect
Yes.
by the chromatin. Right?
By the centromere.
Centromere, I
And
mean.
that creates a tetra. So remember, during interphase of all cells, you have a chromosome 1 and it duplicates to make a sister chromatid, right?
Yeah.
Okay, so this is the sister chromatid, that one yellow thing. Okay, you have 2 branches of identical stuff. Okay,
So like first you have those homologous pairs are when like you have one from your mother, one from your dad.
and they connect. They make this is a homologous pair. This is a homolog. So okay, you learn the names later. Just understand the process first. So remember, during interphase, you had every cell had one from the mother, one from the father, and they duplicated. So you have sister chromatids, meaning identical, identical. And then during meiosis, you're using those genetics, like each chromosome one, to come together and create something called a tetrad. And when this tetrad occurs, there's overlapping, and genetic information can be switched. And that's crossing over. So this happens in prophase 1. So crossing over, homologous chromosomes exchange corresponding segments. And remember, if you're giving up like the same amount of like 4 segments, you have to take 4 segments. It has to be evil. If it isn't, then that's a mutation, can cause complications. Okay, chromosomes condense further, spindle fibers form, tetrads move to the center. So you have like, remember, if you have 23 chromosomes, you have 23 tetrads, right? Because you've got, and each one, so you have 46 sister chromatids. So you have 2, 2, 2, 2. Okay? Is that clear? Okay. The next thing is metaphase 1. Okay? The tetrads align at the center. Remember, metaphase, middle. So that's all that happens. Yeah. The spindle fibers, or what do you also call them? There's another name to it. Oh, they're called spindle fibers. They attach to these chromosomes to the centromeres of the chromosomes from either poles, because your poles like the North Pole, South Pole of the world, and that they're going to pull them apart. Now, anaphase is the most important part, because anaphase is very, very different from mitosis. Because what happens is normally in mitosis, if you can see,
They just split up.
yeah, they would split up, and you would have identical on each side, and it'd be diploid, 46, 46, you know? But instead of what happens in mitosis in anaphase 1, sister chromatids move as pairs instead of splitting up. Meaning, if I have 2 from the mother, 2 from the father, ideally, 1 from the mother should go, 1 from the mother should go there, 1 from the father should go there, 1 from the father should go there. But instead, here, it splits up like that. And normally, it would be completely different, because one would have the mother's information, one would have the father's information.
Crossing over occurred.
Yeah, but because of that, you have a mix of both your parents.
Yeah.
So that's the key thing. That's why crossing over is so crucial in meiosis. Otherwise, you'd look identical to your mother or identical to your father, depending on, except for the fact that the sperm and egg meet, and then you get. So you can't get any of your, if you get all your grandfather's information, you wouldn't get your grandmother's information unless crossing over occurred. Does that make sense?
Yeah.
Okay, so that is when the ploidy number goes from 2 end to end. That's the most important thing. And so if anybody asks you, in which meiosis and which step does the ploidy number drop, you have to say anaphase 1. And remember to put the 1 on which you don't get credit. I've made that mistake so many times. So that's the most important thing, okay? So they're separated.
But why does it drop?
Because remember, like, let's. Can I have a piece of paper if you don't mind?
Yeah. You can just do it.
Yeah, okay. So you know how we have, like, I have chromosome 1 from my father, chromosome 1 from my mother? So in a normal, like, mitosis, what would happen was it would spread up. So I'd have mom, dad, both of these being identical, and one would go there, one would go there, one would go there, one would go there. So I'd have mom and dad. But what happens is during mitosis, this in meiosis and anaphase 1, the entire thing goes. The entire thing goes. So technically, you only have one number 1 chromosome, which means that it's a haploid, not a diploid. Does that make sense?
Kind of.
Like, think about it like here in mitosis, we're splitting them up, so you still have genetic information.
Identical.
Yeah. But for this one, because you're splitting it in half, it's not it's not 2 parents anymore, it's just 1 parent's information. So that can cause, that means that you're dropping the haploid number.
Yeah.
I mean, dropping the ploidy number to haploid. Okay. The next one is telophase, and telophase 1 is pretty normal. It's only haploid on each pole. And okay, so look here.
So can you like explain haploid?
Okay, you know how all the cells in your body have 46 chromosomes.
Yeah.
23 from your mother, 23 from your father.
Yeah.
But your eggs, the ones that have come from germ cells, only have, instead of 46 chromosomes, only have 23.
Yeah.
And what happens is, the reason they have 23 is because
I need the guy
Yeah.
23.
Exactly.
Yeah.
And if you had 46, then you wouldn't have a baby anymore because you'd have a 3N ploidy number, and
Yeah.
that's not you.
Yeah.
Like, for example, I think it's frogs that have a 4N ploidy number. That's
Yeah.
just
So like 46 and 46.
Yeah.
Okay.
So for them it's like, I think they need.
So like their egg cell, so the girl's egg cells has 46 chromosomes, so that's 23.
No. Yeah,
Yeah.
23 out of 46. Your egg has 23 chromosomes,
Yeah.
and his sperm will have 23 chromosomes, and then the zygote will have 46.
46. Yeah.
But all the cells in our body have 46 chromosomes.
Mhm.
23 from our mother, 23 from our father.
Uh-huh.
Does that make sense? And
So like haploid is
23,
and then
diploid is like double, 2 and 46. And also, if you think about it, when you do mitosis, you need all those chromosomes because you're not getting any more. So that's why mitosis is identical.
Yeah.
Okay. During telophase, you have N on each pole, and if you look here, I tried hard to draw. I showed you, like, in mitosis, you're getting half of each for each parent and each chromosome.
Mhm.
But from mitosis, you're only getting one chromosome, number 2 chromosome, number 2 chromosome, instead of this way.
Mhm.
So here you only have 23, here you have 46. Okay, the nuclear envelope starts to regenerate, cytokinesis occurs, and you're left with 2N.
Yeah. 23.
Yeah.
No, 46.
Okay, then we move on to meiosis 2. During prophase, the same thing happens.
Wait, so it creates 2 haploid cells?
Yes.
Okay.
For meiosis.
Yeah.
And remember, look, if you've noticed, over here you only have one chromosome, but here you're still left with sister chromatids.
Yeah.
Meaning we can break this down even further. That's why we go into meiosis too. And so in prophase, the same thing happens. Spindle fibers form, chromosomes move to the center, nuclear envelope starts to degrade or break down. Then comes metaphase 2. And in metaphase 2,
That's when they finally
you split them into single chromosomes, into one strand instead of sister chromatids. And this is the other name, spindle fibers, microtubules. They attach to the centromeres and they pull the sister chromatids apart. And then in telophase, you're left with 4 different haploid cells. And that's all. Does this help?
Yeah.
Do you have any questions?
When do you call a chromosome chromatid?
That's the tricky thing. Chromatids, okay, chromatin is all the chromosomes, okay, and when it condenses, they're all chromosomes. That's the thing. It's very complicated because you know how when you say, oh, I have a cell, these are all the chromosomes? That's correct. When you refer to the one strand, it's a chromosome. When you refer to sister chromatids, it's still chromosomes. So that's very complicated. Just know that sister chromatids, are the duplicated chromosomes. So you have 2 identical duplicated strands that are created in.
But then, so these would be in metaphase. So these would be considered sister chromatids in telophase.
Yes.
But then they're not identical.
No, those 2 are. This was never split in half.
But then crossing over occurred. In
The crossing over occurred, so that's like a mutation. But sister chromatids are supposed to be identical.
But then how are they identical if crossing over occurred?
No, like initially when they're created, they're just a duplicated version of the chromosome. That's what the sister chromatids by definition are 2 identical strands from the same parent. And then when crossing over occurs, they're not identical anymore, but they still have the same structure because they're still chromosome number 1.
Yeah.
So sister chromatids initially when they're created
Are identical.
Yeah, when are they created? When does it duplicate? What comes before mitosis and meiosis?
Oh yeah.
Both of them. Interphase. Okay? I didn't cover interphase. Interphase is just like, the gap here and everything.
Yeah.
Sorry, gap phase. And when that's what I'm thinking about all the time. When the chromosome duplicates, it has 2 and it's connected by a centromere. And when it's connected by the centromere, that's the sister chromosome. And in mitosis, the sister chromatid is split
Yeah.
into two.
Yeah.
But here, it's only in metaphase 2 that it splits. And that's why
Yeah.
meiosis 2 is identical to mitosis.
Mm.
It's really like, but would you guys like me to photocopy these? Okay, and then that way, like, honestly, the best thing to do is when you read, write it down in step-to-step form.
And then like draw it out.
Yeah. So, and you know, people are like, oh, I just memorized it. But what happens is if they ask you like a application question,
Yeah.
like where does it happen?
Yeah.
They don't know because they, you know, you
Yeah.
have to like, so it's, but then you have to memorize a few things. Like know that it's an anaphase one that the protein does. Okay. Anything else?
Nope.
Okay.
Can you
No
photocopy
problem.
it? And then,
Yes.
are you gonna go to study hall?
Yeah, I'll
Okay.
bring it to you.
Okay.
Actually, I might not come, but I'll still bring it to you.
Okay.